We are going to begin our discussion on genetics by talking about this gentleman here, Gregor Mendel. So Gregor Mendel was really interested in heredity, which is the transmission of traits from one generation to the next. Okay. So he began to run some experiments. So before Mendel came around, the general thought was that we were all a blend of our parents. So if we are going to use plants, which is what Mendel used, a red flower plant and a white flower plant would have pink flowered plants. Okay, so you would be basically a exact blend of your parents. All right, which we know that isn't true. Sometimes people look really much like their dad or really much like their mom or nothing like either one of them, depending on how things worked out genetically. So, but prior to Mendel, this is what they thought. So Mendel used pea plants like green peas, and he traced traits from one generation to the next. So his thought was, or his hypothesis was that there are different versions of a gene, which he called alleles, and that those determine heritable traits. So the first thing he did was he identified some characteristics that he was going to study. All right, so we had flower color, purple and white, flower position, was it on the axial or on the top? Seed color, yellow or green, round or wrinkled. So he was studying all of these characteristics, sometimes together, sometimes separately, in these pea plants. Now, in order to talk about what happened, we need a little bit of vocabulary. So let's get some. First of all, he developed 4 hypotheses, all right? Number 1, there are alternative versions of genes. That's the definition of allele. Alleles are alternative versions of alleles that account for variations in inherited characteristics. So these alternative versions or these alleles are what makes every plant and human being sort of unique. All right? Now, for each characteristic, which is flower color is the characteristic, the trait is yellow or green, I think. So for each characteristic, an organism is going to inherit 2 alleles of a gene. So you have 2 versions, say, of the widow's peak, which is right here. I have a widow's peak. Some people have flat. I have a widow's peak. Okay. That is genetic. So I got 1 allele from my mom and 1 allele from my dad. Now, if my mom and my dad both had the exact same trait, so they both had widow peaks, we would be homozygous. Homo means same. All right. So we would have the same allele from both parents. Now, if my parents did not have the same allele, we say that it's heterozygous, and that means it's different, okay, for each gene. So these vocabulary words are so important as we move forward. All right, if the 2 alleles are different, one of them is going to win. All right, so if I have no widow's peak and widow's peak allele, so no widow's peak from mom, widow's peak from dad, one of those has to win. All right, that's called the dominant allele. That's the one that wins. We'll say the one that wins. So if you only need one of them to win. All right, so, oh goodness. So only need 1 to win. So if the dominant allele is there, it wins. But if you have 2 different ones, one is dominant, the other one is recessive. In order for the recessive allele to show, you have to have 2 copies. So in order for the recessive allele to win, you cannot have any dominant alleles present. Okay? Now, we usually indicate a dominant allele. So if we were going to do, say, widow's peak, which I would do a big W for dominant and little w for recessive. So usually recessive is the lowercase letter and the dominant is an uppercase level. Yes, letter. A sperm or an egg carries one allele for each inherited character because allele pairs separate during gametes, during the production of gametes, which is meiosis. All right. So when we undergo meiosis, we separate out the alleles so that there's only one copy of each characteristic inherited in a sperm or in an egg. Now, because of these experiments, which we're going to walk through in just a second, the blending hypothesis is rejected because what the blending hypothesis doesn't do is explain how traits can skip a generation. So we're going to sort of walk through how that might happen. All right, so the experiment. Mendel started off with 2 pure breeding plants. So if we're doing flowers, pure breeding means they're true breeding means they're homozygous for the trait. So this pure breeding guy, we'll say I think purple's dominant. So big F's for purple. And then we have our white flowers, little f's. Okay. And that's true breeding. And P is the parent generation. Now, the kids of these 2 flowers were hybrids, but they were all purple. Now, remember I said this is FF and this is FF and this is F, little f, little f. So the only thing that this, let's say egg can have is a big F. And the only thing this sperm can have is a little f. So these are the F1 generation, or the first filial generation is a hybrid, or we call it heterozygous, because it has one big letter and one little letter. All right. Now, what happens when we take 2 of these heterozygotes, so big F, little f, and we cross them? This is where it gets interesting. So not some of the plants become white. Now, this generation had no white plants, none. Okay. But then all of a sudden in the second generation, we have white plants reappear. Okay. Now, how does this happen? Well, we're going to get into Punnett squares in just a little bit, and we're going to explain why it happens. But first, we need to talk about probability. All right. Because when we do our crosses, this is what this is a cross, we are going to have to come up with the probability that something will happen. Okay. Now, one of the reasons I don't like this example is because they show 3 plants being this and 1 plant being that. And that is not how probability works. So let's talk about probability really quickly before you move on to the next video regarding how we do these crosses. All right, so Mendel's laws reflect the rules of probability. The rule of multiplication means that the probability of 2 independent events occur, you multiply. Division calculates the probability of an event that can include in alternative ways. So don't worry about that question. We're not getting to that in a second. So if we go back, if we're doing this rule, well, first let's look at our coin flip. So if you flip a coin, your alternatives are heads or tails, correct? So if on heads, I put a big B, and on tails, I put a little b, okay, then what is going to happen is that the female, so in the coin flip of a female, she can either give a big B or a little b because she's heterozygous, right? The male can have a sperm with a big B or a little b, okay? Because this, you have a 50% chance of getting a sperm with this and a 50% chance of getting a sperm with that, okay? Now, if we plot these, this is an example of a Punnett square. Then what we have is we have a 50% chance of getting big B or little b from mom, and a 50% chance of getting big B, little b. Oh, I got them wrong. This is mom, this is dad. But we have 50% chance of each. Now, we combine them in any sort of way. So for instance, this big B can combine with this other big B, big B, big B. And because of the rule of multiplication, we multiply 1/2 by 1/2. We have 1/4 of a chance of having a homozygous dominant individual because big B is homozygous dominant. All right. Now, if we bring this big B down right here and we pair it with a little b, we have a 1/4 chance, again, rule of multiplication, of having this hybrid or heterozygous. Now, if we look over here, if we take the big B from mom and combine it with the little b from dad, we have another 1/4 chance. Here's where addition comes in. Look at this. I need to add my 1/4 chance here and my 1/4 chance here because this is the same, big B, little b. So big B, little b is heterozygous. And I am going to add 1/4 and 1/4. And so I will have a 0.5% chance. That's the law of addition. And then we have little b, and because of the law of multiplication, we have a little b homozygous. All right. So this is a little bit of math, a little bit of probability, but this is going to come in handy for you to understand this as we move forward to what we basically did right here, which is a monohybrid cross.